Hi everyone, welcome to this very first EpIC seminar of 2024. So today we're really pleased Dr. Alex Miller. So Alex, I think most of you probably have come across in the field because she's worked in Ion Channel since the very beginning of her academic career. So she did her PhD with Stephen Long at Sloan Kettering, where she worked on 2,4-potassium channels and bestrophin ion channels. And after that, she went to work with David Clapham at the HMI. She's won a number of prestigious awards, including the Leading Edge Award and the NIH Future Research Leader Award. And she's recently started her own group at the University of Iowa, where she's focusing on viral ion channels. And this is some of the work that she'll tell us about today. So without further ado, please welcome Alex.
All right, thank you. I'm just gonna share some screen now. Yes, well, thank you, Layla, for that introduction. And I also, I thought I would make a point to thank the EpIC committee for all of their work toward this and for launching the seminar series, because I really do think it provides a great platform for late-stage postdocs and young investigators to present their work to the SGP community. And it's been a terrific series so far, what I've watched and what I've watched by recording. So, thank you for providing this for the community. Okay, so as Layla mentioned, I moved to the University of Iowa a few months ago, and I'm actually in the process of setting up my lab. So what I'm actually going to be sharing with you today is our work to characterize the structure and function of the SARS-CoV-2 protein OR3A, which is actually a project that I pursued during my time in David Clapham's group as a postdoc. So since my background is in primarily ion channel structural biology research, I was really interested in this project since OR3A was previously reported in the SARS literature to function as an ion channel. However, I hope that I can convince you today, in fact, that SARS-CoV-2 OR3A is not an ion channel, but it instead likely contributes to SARS-CoV-2 mediated cell endocytic trafficking defects that are seen. All right, so taking a step back for a minute to briefly touch on the topic of ion channels, which I'm aware we're all very familiar with in this crowd. I find these proteins really neat because they're conceptually very simple in their function. Maybe I'm oversimplifying it, but they form holes that span membranes, yet they're fundamental for rapid physiological responses, such as muscle contraction, action potential conduction, synaptic transmission, et cetera, right? And the three key features that are shared by all ion channels, as we know, is that they allow for permeation. They can discriminate between ions of different size or charge, and they undergo conformational changes that open and close the pore called gating. So I bring this up because the question is, could ion channels also be part of the viral armamentarium? And yes, they are known to encode for proteins that form channels. However, they're not channels in the classical sense as I just described, because in most cases, they are not selective for an ion or a particular series of ions, although there are exceptions to this. And many are not gated. And so for this reason, these are typically called viroporins. Again, viroporins are membrane proteins that form holes that are permeable to ions and often actually small molecules. And here on the right are three examples of viroporin structures. You can see my cursor, correct? I hope so. The M2 protein from influenza A. This is the VPU protein from HIV, and the P7 protein from hepatitis C. And what I hope that you can appreciate from this is that each forms an oligomer with a hydrophilic hole or pore within the middle of the oligomer, which one wouldn't anticipate if it's a channel. So, the big question is, why do viruses need these, right? And well, generally proteins encoded by viruses, will function to promote their survival and propagation in the host in some fashion. So some viroporins, including the M2 protein from influenza, achieve this by subverting the host cell trafficking machinery. So M2 protein is actually quite important for cell entry. But in general, I would argue that a mechanistic understanding for many of these putative viroporins is not well described or defined. So, all right, so work from the SARS literature has suggested, as I mentioned on my first slide, that SARS-CoV-2 or3A may be a viroporin. But at the time we started this project, there was no structural information available, and that's part of the reason why I was interested in this. Okay, so or3A is one of 29 proteins that are encoded from the SARS-CoV-2 viral genome. It's among a subgroup of proteins that are called accessory factors, which are in green here. This is a 3A. Many of which of these, at the time when we started this, were poorly characterized proteins compared with the non-structural proteins, or the NSPs in blue here, in which a large subset of these are involved in RNA replication, and the structural proteins in red here, which form the virion. And these are the spike protein, which we're all familiar with, the envelope protein, the E protein, the membrane M protein, and the nucleocapsid protein that's in here, the N protein. Okay, so what is our knowledge of the SARS-CoV-2 viral life cycle? So I figured I'd just walk us through it, and this is a very simplified, model. And potentially a bit outdated at this point, but it gets the point across. Okay, so SARS-CoV-2 enters the host cell by engaging with the host cell receptor ACE2. And a number of events prime SARS-CoV-2 for viral entry, either directly by fusion to the plasma membrane or through the endocytic pathway. But in either case, the RNA genome must be released into the cytosol in order to be translated by the host cell machinery to make components of what's called the RNA-dependent RNA polymerase complex, or RDRP complex. For some of the, the non-structural proteins that I showed you earlier, that's what these are the components that make up that complex. And so the assembly of the RDRP complex is necessary for the next steps of synthesizing the RNA genome for viral progeny and transcription of the latter part of the genome for synthesis of the accessory and the structural proteins. So the current model is that this is done in the area of the ER that's been actually hijacked by the virus, and which forms these convoluted membrane or double membrane vesicles. And this is proposed to act as a shield to protect viral RNA from host cell degradation. That's at least one idea. Next, the subgenomic mRNAs, as I mentioned, are translated into the structural and accessory proteins. Again, the structural proteins form the virion, and the assembly occurs in a region called the ER-Golgi intermediary complex, or the ERGIC. And so in this diagram, you can see the spike protein, the envelope protein, and the membrane proteins coding the ER-Golgi intermediary complex. Now, the fourth protein in the structural protein, which is this nucleocapsid protein, needs to assemble with and bind to the viral progeny RNA. And this complex buds into the ER-Golgi intermediary complex and is encapsulated to form the virion, which then is by some mechanism trafficked out of the cell. All right, so many of the accessory proteins, as I mentioned, at least when we started this project, were poorly characterized functionally, including SARS-CoV-2 or 3A. So, what could be or3A's contribution to SARS-CoV-2 pathogenicity? It's an outstanding question, but if it participates, for instance, in the viral life cycle, it would be likely involved in the later stage of this process, such as viral assembly or viral exit, just simply because it's translated during or after progeny RNA replication. So it's just something to keep in mind in terms of understanding its function. And so determining if ORF3A may function as a viroporin would better help elucidate its contributions during COVID-2 infection. And so we set out to answer this question by taking a simple two-pronged approach of functional characterization combined with protein structural determination. And so to test for viroporin or channel activity, we took a variety of different approaches, including patch lamp electrophysiology and the development of cation and anion transport assays. And then for protein structural determination, we turned to cryo-EM. Now, for both structure and function, having insight as to where OR3A is localized in the host cell is really important, not only to identify the area of the cell to patch, for instance, but also to guide OR3A protein sample preparation for cryo-EM, because one would ideally like to try and mimic the lipid environment that the protein may natively reside in. And so the first question we asked was, where is OR3A localized in the host cell? And to answer this question, I generated a doxy-inducible stable cell line of OR3A fused to a halo tag, and then used fluorescent proteins that are targeted to different subcellular compartments. And what we found is that OR3 is localized at the plasma membrane, and it's also localized in the endocytic pathway. And so here are a few examples where OR3 is co-localized with a farnesylated GFP marker for the plasma membrane, as well as a marker for early endosomes, RAB5, and a marker for lysosomes, LAMP1. Okay, so not only have we observed this pattern, but at least 4 other groups have published similar data that agrees with our results. And in particular, in the Gordon et al. Paper, they not only demonstrated this by overexpression of OR3A, they actually also demonstrated this during CoV-2 infection, so by staining cells that were infected with SARS-CoV-2. So this really suggests to us that our overexpression data is consistent and likely physiologically relevant. Okay, so now that we know where OR3A is localized, we then performed patch clamp, whole cell patch clamp, I should say, and so at the plasma membrane, and also in endolysosomes and AGK cells. And so our take-home message from these experiments, unfortunately, is that we do not observe a significant OR3A attributable current in either compartment. All right, so here's an example of our experiment, our whole cell patch clamp experiments. So with an overexpression of OR3A, we see a fairly negligible difference in current density in a variety of different external cation conditions that we tested. And so the figure here on the left is just showing the current density measurements at -80 and +80 millivolts in cells that were untransfected or cells overexpressing CoV-2 OR3A, and that's in red and pink here, recorded in an extracellular sodium, potassium, or cesium chloride. And then on the right here is a similar experiment with a larger range of external cation solutions tested, including calcium and the large inorganic cation NMDG. And so all of this is to demonstrate that we do not observe OR3A attributable currents above background, at least at the plasma membrane. Okay, likewise, with endolysosomes containing OR3A, which here are represented by the green trace, okay, we observed little contribution of potassium or sodium in this solutions, the experimental setup here on the left, or calcium in this experimental setup on the right, compared to the background endolysosomal currents that were measured in black. And I'm not showing this here, but it's in our paper, you can check it out. But we also tested this with a chloride and protons, and we were not able to measure any attributable currents there. Okay, so then we thought, could we be missing something? And so we asked, whether we could mimic the conditions that were used to characterize the original data on SARS OR3A, or I'm going to now actually refer to this as SARS-CoV-1 OR3A. And those were 2 electrode voltage clamp studies. So those were done in oocytes. And so we investigated the channel activity of SARS-CoV-2 OR3A in oocytes. And again, we weren't able to measure any OR3 attributable currents above background by TEVC, and similar extracellular solutions that were published previously. So this would be around 96 millimolar potassium chloride, or even in standard TEVC recording conditions containing 96 millimolar sodium chloride. Okay, so all of the evidence that I've shown you so far points to SARS-CoV-2 OR3A not functioning as an ion channel. Concurrently, we asked whether the atomic resolution structure OR3A is consistent with it being a viroporin. And so for this, we took advantage of cryo-EM. All right, just give me a sec. All right, so the first experiment that I did back in April 2020 was to evaluate what the oligomeric state of OR3A is. In the SARS literature, OR3A was predicted to have 3 transmembrane helices and was reported to be either a homodimer or homotetramer. I guess it could be both, actually. But cross-linking a folded intact CoB2 OR3A from isolated cell membranes appears to migrate as a dimer with increasing concentrations of a cross-linker. Similarly, purified OR3A elutes by size exclusion chromatography in a detergent micelle at a volume that is consistent with it being a dimer. So as a dimer, the molecular weight of OR3A is about 64 kilodaltons, which is very small for cryo-EM structural determination, and especially for membrane proteins. So this made it a fairly challenging project, at least for me. Nevertheless, I prepared nanodiscs that contain a mixture of lipids that mimic the late endosomal lysosomal membrane and reconstituted the protein, and we were actually able to determine a structure of SARS-CoV-2 OR3A. And so here is an example. This is the overall cryo-EM density for one of the structures that we determined, again, in this late endosomal lysosomal lipid-like environment. And this is using a standard scaffold protein called MSP1D1 for formation of nanodiscs. And this structure was determined at 3 angstrom resolution, but we did determine some other structures, one of which was at a higher resolution. I think it was 2.8. And that was in a similar condition, but using a different scaffold protein called saposin A to form the nanodisc. Anyhow, and here is the SARS-CoV-2 OR3A model with 3 views facing the membrane. This is the extracellular or luminal side above, and the intracellular side below. Okay, so OR3A is a homodimer, as we expected from our experiments that I showed on the previous slide. There are 3 transmembrane helices provided by each subunit. And that's, you can see here is TM1, 2, and 3, followed by a structured cytosolic domain. So in addition to the protein density here, we were also able to resolve at this resolution 2 non-protein densities between the dimer interface, which is in orange here, and in a membrane exposed group between TM2 and TM3, which is here in purple here. And we believe that those are likely 2 lipid molecules, and so we assign these as DOPE molecules. And I'll add here that while we were working on this project, Steve Rohan's group at UC Berkeley solved a very high resolution structure of SARS-CoV-2 OR3A, and they were able to resolve this at 2.1 angstrom resolution, which was really quite a feat given how small the protein is. And they were partially able to achieve this with collaboration with Thermo Fisher, because they had used their latest cryo-EM equipment, including a cold FEG and a Selectress energy filter. But I thought that was really cool. Anyway, so if OR3A is a channel, where is its ion pore located in the structure, right? That's the big question. So we inspected the transmembrane region for an aqueous cavity, but approximately 2/3 of it is tightly packed in its current conformational state, which is depicted in white here and here. With the narrowest point of this region, which is less than an angstrom in radius, which would be too narrow to accommodate a dehydrated cation. So this indicated to us that either the structure represents a non-conductive or closed state of OR3A, so we can't rule out that it isn't a channel from this, of course, but it is possible as well that it is not a channel, right? However, we did observe an aqueous vestibule that extends beyond or into the cytosol, I should say, and that's colored in blue here. And we asked what are the residues that line this vestibule, and what we noticed is that there were 10 positively charged residues that are contributed by both pronomers. And this actually creates a positively charged region that would likely not be favorable for a cation channel, which is what OR3A was reported to function as in the SARS literature. And so our structural data in combination with our patch clamp electrophysiology experiments continue to support the hypothesis that SARS-CoV-2 OR3A is not a viroporin. So we further explored this question by testing purified and liposome reconstituted SARS-CoV-2 OR3A in a variety of different functional assays, including cation and anion transport assays, as well as proteoliposome patch clamp electrophysiology. All right, so I'll briefly describe the transport assays that we use to test OR3A. These are just typical ACMA-based assays. So the idea is that, and so I'll be brief. So a potassium or chloride gradient is established with the vesicles that have been reconstituted with your protein of interest. So in this case, OR3A. And if OR3A is a functional channel, then potassium or chloride should be transported in these conditions. And the readout of this assay is a loss of fluorescence over time with the binding of protons to ACMA. But so if the channel is closed or blocked or inactive in some way, or if you're using, let's say, a vesicle only control, then there should be minimal loss of fluorescence. And so that's what the readout is. And so what we see is that OR3A does not transport potassium, and it doesn't seem to transport chloride either, compared to the vesicle only control in black. And just to add, for the potassium transport experiment, to control for the total vesicles in the preparation, we added a potassium ionophore valinomycin at the end of the experiment. Okay, so in these experiments, and this is pretty critical, we reconstituted OR3A into vesicles using a typical weight-to-weight ratio of channel to lipids of 1 to 100. And it really does matter, and you'll see why in the next few slides. Okay, so we also asked this question using proteoliposome patch clamp, and we similarly were unable to measure significant currents, or I should say conductances, I'm sorry, in this case, attributable to SARS-CoV-2 OR3A in symmetrical conditions containing calcium, potassium, or sodium cations. All right, so all of our data so far is consistent with SARS-CoV-2 OR3A not being an ion channel. But of course, I'm describing one side of a story here. And I mentioned that this is a protein that's been annotated in the literature to function as a viroporn by a few groups, actually. And so I thought it would be helpful to provide at least one example of why we think this may be a case of mistaken identity. And so when a putative channel has been purified and reconstituted for characterization by either proteoliposome patch clamp or by, let's say in planar lipid bilayers, it's important to remember a couple of things, right? First of all, that patch clamp recordings, detect single proteins, right? They're single channel openings, and thus, far exceed the limits of specific protein biochemical purification. So in other words, a common concern is that a current that one is recording may not be your channel of interest, but that's, I guess what's come along for the biochemical ride and as a contaminant. Secondly, transient membrane leak is a common phenomenon when recording at high protein concentrations. So an unfortunate recent example of this problem occurred, I guess, in the last few years, which was the characterization of a putative mechanosensitive ion channel called Tecan, which has since been demonstrated by a number of groups now that not to be a channel. And so, we asked if either of these points could be why SARS-CoV-2 OR3A was misidentified. And so to test this, we first reconstituted OR3A using a high protein concentration and perform proteoliposome patch clamps. So now instead of using this 1 to 100 weight to weight ratio, we use 1 to 10. And when we did this, we were actually able to detect multiple calcium, sodium, and potassium conductance species with varying open probabilities. So here is on the left are some exemplary gap-free recordings. And the downward deflections, here represent channel opening events. And on the right is a summary of our data with, and you can see the multiple conductance species that we observe. And that's quite, different to what I showed you previously when reconstituting the protein at a 1 to 100 lipid ratio. So again, probably don't need to explain this to this crowd, but, observing multiple conductance species, 4 in the case of sodium, 7 in the case of calcium, is highly atypical of an ion channel. Normally, channels have a signature conductance. They open and close. They may have a subconductance state couple, but the C7 conductance species is very odd. Furthermore, it was odd that we observed single channel events from a sample containing an abnormally high concentration of protein, because based on the average diameter, we averaged a guessed of a patch. We estimate that each patch should have contained on the order of hundreds of channels, based on this protein to lipid ratio. And that honestly is a conservative estimate. And so we are, in addition to that, I should say, we also did not observe, any potassium flux in this sample. So by the potassium flux assay, which is an indirect measure of macroscopic activity. So, all of this is to suggest to us that the conductances that we measure are likely due to, or could be due to transient membrane leak, and are likely spurious currents. However, another possibility, and of course, this isn't mutually exclusive, is that the current could be contributed by a contaminating channel, which isn't detected in the flux assay. So to address this latter point, we actually went to the trouble of performing mass spec with the ORF3A reconstituted liposomes. And so from this list of proteins identified in the sample, we sorted this list to look for membrane proteins that contain 2 transmembrane helices or more, which is a likely requirement for ion channels, and found that among the top 5 hits, 2 proteins that appeared were VDAC1 and 2. Okay, so then we wondered, based on this data, if a VDAC channel could be contributing some of the conductances that we observed by proteoliposome patch clamp. And so we performed similar recordings of OR3A and added a blocker. We just used DIDs. And what we were able to see is, in fact, we were able to block some of this current. And so this is an example of the DIDs block to the left here. And then the washout you can see here. And then this is a summary of the channel open probability for 6 traces on the right. And so at least we now believe that, at least in our OR3A reconstitution preparations, that some of the current that we measure in these samples may be attributed to VDAC channels that were purified along with OR3A. All right, so again, the summary here is that we think this is a case of mistaken identity to common experimental problems that arise when characterizing putative ion channels. However, I guess one question remains, and that question is, could SARS-CoV-1 OR3A be evolutionarily distinct from SARS-CoV-2 OR3A, where, let's say, the latter has lost its ability to function as an ion channel, but SARS-CoV-1 OR3A is still an ion channel, right? Or, is SARS-CoV-1 OR3A misannotated in the SARS literature? And so to answer this question, we took a similar approach of testing SARS-CoV-1 OR3A by whole cell patch clamp, by TVC, to directly compare our results to what was previously published, and also by determining its structure. So SARS-CoV-1 and SARS-CoV-2 OR3A are 73% identical in sequence to one another. And apart from these differences, the overall architecture of SARS-CoV-1 OR3A looks nearly identical to what we determined for SARS-CoV-2 OR3A. So on the left here is a superpositioning of SARS-CoV-1 and SARS-CoV-2 OR3A. And the overall architecture appears to be the same. You can see that the global RMSD is less than 0.5. And on the right-hand side here is the structure of SARS-CoV-1 OR3A with the whole diagram superimposed, again, just to demonstrate that this region would not be able to conduct a dehydrated cation in its current conformation. And I don't have it here, but it also harbors that same basic aqueous vestibule here. So it looks very similar to SARS-CoV-2 or 3A. And so the structure alone is already suggesting to us that it's likely not a viroporin. But what about, what do we gain from the electrophysiology data? Well, again, we were not able to see any attributable currents for SARS-CoV-1 or 3A by whole cell patch clamp, comparing IV traces from HEK cells that were untransfected in black, transfected with CoV-2 or 3A in red, and then transfected with a CoV-1 or 3A in blue, and also by 2 electrode voltage clamp. Okay, so we conclude from our work that OR3A proteins are not viroporins, and they're misannotated in the SARS and COVID-19 literature, which, you know, I view as an important conclusion that changes current models of how this accessory protein may contribute to viral pathogenesis. I hope you agree. All right, so if, you know, OR3 proteins are not ion channels, then the big question is, what is their function, right? So I get asked this question a lot, and, you know, we asked ourselves this question as well, and it's a big question, right? But one way to approach this is to ask, does SARS-CoV-2 or 3A interact with any host proteins, and could this shed light on its function? And so a number of studies have been done to address this question by traditional IP and mass spec, and as well as by using proximity labeling strategies such as EpIC and BioID, et cetera. And the first large-scale study performed was a simple experiment of overexpression each of these SARS-CoV-2 proteins in HEK cells, followed by an IP with the CoV-2 protein and then mass spec. And so in this study, they identified 8 host proteins that appear to interact with OR3A, and 2 of these proteins form part of what's called the Hops complex. Okay, so what is the Hops complex? Well, as cleverly stated in the title, it's a vesicle tethering complex that mediates the fusion of autophagosomes with late endosomes and lysosomes, and also late endosomes with lysosomes. So if SARS-CoV-2 or 3A interacts with VPS39 and VPS11, this is a very old model of the Hops complex from negative stain cryo-EM data. One hypothesis that that is that it may impair Hops complex activity if it, you know, and that would basically disrupt a vesicle fusion in that case. So we asked if, you know, if we overexpress OR3A, do we see a defect in late endosome and autophagosome vesicle fusion? And in fact, we do. So using the same doxy-inducible stable cell line of OR3A fused to a halo tag, again, in magenta, and that's in magenta here, we stained for a marker of the late endosomes RAB7 in green. And as you can see, there is an enrichment of RAB7 containing vesicles when OR3A is overexpressed, suggesting a defect in fusion. And we've also actually observed a similar phenotype when we've used a marker for autophagosomes, which is called LC3. So that's interesting. And then we asked, does VPS39 and or VPS11 interact with OR3A? And right. So although we didn't observe any interaction with VPS11, although I'm not presenting that data, we did observe an interaction with VPS39. So in this experiment, we used detergent purified CoV-2 or 3A, and we mixed it with cell lysate containing overexpressed VPS39 GFP. And so what we did is we IP'd for OR3A and then blotted for VPS39 GFP. And I hope you can see, just pay attention to lane D. That's the relevant one in this case. That VPS39 is being pulled down with increasing concentrations of purified CoV-2 or 3A. So the higher the concentration of CoV-2 or 3A, the more VPS39 we're seeing by Western at least. So this was exciting to confirm, you know, to see this interaction, and that it corroborates with and that it corroborates with this RAB7 accumulation phenotype. Okay, so then the next question that we asked is, is SARS-CoV-1 or 3A can also co-IP VPS39? And actually, to our surprise, it does not. So it doesn't appear to be interacting with VPS39, even using this highest concentration of purified CoV-1 or 3A that we used in this in the assay up here. And likewise, SARS-CoV-1 overexpression does not produce the Rab7 accumulation phenotype that we observe for SARS-CoV-2 or 3A. And so, this is actually quite remarkable because it suggests that this phenotype may have been acquired in SARS-CoV-2. I thought that was neat. So since we observe an interaction of EPS39 with SARS-CoV-2 or 3A, but not SARS-CoV-1 or 3A, we asked if we could identify the site of protein-protein interaction. And so by inspecting the cytosolic domain of both OR3A cryo-EM structures, and by pinpointing the divergent regions of amino acid sequence conservation, we identified an unstructured loop that could be facilitating this interaction. And that's in yellow here. And I guess say partially unstructured because you can see a number of the residues are modeled in. But to answer this question, we generated loop chimeras of SARS-CoV-1 and SARS-CoV-2 or 3A, substituting the SARS-CoV-2 loop with SARS-CoV-1 or 3A here. And likewise, we generated the SARS-CoV-1 or 3A chimera containing a SARS-CoV-2 loop. And so the first question we asked is, if we substitute the CoV-1 loop into SARS-CoV-2 or 3A, will this reduce its binding with VPS39? And indeed it does. So this is, again, using increasing concentrations of CoV-2 or 3A. This is the wild type, and this is the loop chimera. We see in lane D a loss of, or a reduction of interaction with VPS39, especially when compared to the control. And likewise, we asked if the chimera might alleviate that Rab7 accumulation phenotype that we originally observed by overexpression of SARS-CoV-2 or 3A, and it does. So that was great. So this data suggested that the unstructured loop of SARS-CoV-2 or 3A appears to contribute to VPS39 binding, and that this Rab7 accumulation effect is, it may be caused by this interaction. So if this is the case, then we might expect to see the converse, right? To see an enhanced binding of VPS39 to a CoV-1 or 3A chimera that contains the CoV-2 or 3A unstructured loop. And indeed we do. So compared to wild type CoV-1 or 3A, again, pay attention to lane D, we do start to see an enrichment of VPS39 when we swap out and put in the SARS-CoV-2 or 3A loop. However, it's not a full rescue. It's not fully restored, this binding. When comparing it to the relative amounts of VPS39, we're able to pull down with wild type CoV-2 or 3A, right? Especially lane D in this diagram versus this, image. So then we ask, well, do we see this Rab7 accumulation phenotype now that we've introduced the SARS-CoV-2 loop into SARS-CoV-1 or 3A? Unfortunately, we did not. We believe this is likely because the loop itself is not the only region that mediates this interaction with VPS39, and this conclusion is actually supported by another group. So, okay, so with that, let me first summarize what I presented. So again, we asked the question if OR3A proteins from SARS-CoV-1 and SARS-CoV-2 for viroporids, and based on our, I would say, exhaustive work to characterize these proteins as ion channels, complemented with cryo-EM structure, structures for both proteins, we conclude that they're not viroporins, and they're misannotated in the SARS and COVID-19 literature, which again changes current models of how this protein may contribute to viral pathogenesis. Okay, so if OR3 is not a viroporin, then what is its function? Our cell and biochem data suggests that CoV2, but not CoV1 or 3A, interacts with this Hops complex protein VPS39. And in a collection with other publications, we believe that CoV2 or 3A alters Hops-mediated vesicle trafficking by impairing late endosome and autophagosome fusion with lysosomes, right? So, okay, so from a biochemical standpoint, so where is this region of interaction between CoV2 or 3A VPS39? Well, again, we show that the interaction is partially mediated by an unstructured loop of CoV2 or 3A, which is located in its cytosolic domain. And we further inspected this region, comparing CoV1 and CoV2 or 3A structures, and noticed a difference in their surface electrostatics, where CoV1 or 3A appears to be more acidic in this region compared with CoV2 or 3A. And so we wonder if this might be contributing to the differential VPS39 interaction, and if that actually if this may clue us into its full region of interaction. Now we drew two circles suggesting that it's a one-to-one interaction, but we don't actually know this. It could be a 2-to-1 interaction as an example. Okay, but the bigger question here is, why do we care? Like, why is this interaction advantageous to SARS-CoV-2, right? And that's a harder question, but I guess I could speculate for a couple of minutes. In the case of preventing autophagosomal fusion, the first thing that comes to mind is that this mechanism could protect viral RNA and proteins from being degraded, since the function of autophagosomes is to survey the cytosol for abnormal intracellular proteins, damaged organelles, and invading microorganisms, I guess, and then send these components for degradation, right? So I guess that's one possibility. And a second one stems from the idea that beta coronaviruses exit through the endocytic pathway through, I guess, lysosomal exocytosis. And OR3 actually has been implicated in facilitating this process, likely by interacting with membrane tethering complexes that are known to facilitate this type of trafficking, such as BORC, ARL8B. So perhaps sequestering VPS39 might give the virus an additional advantage, as an example, by promoting this type of cellular endocytic trafficking. Anyhow, I think this is overall a fascinating area of biology, and I have several projects and collaborations established to continue to explore this feature in my lab. So with that, I'd like to thank a lot of folks. This is a short and sweet talk. There are a lot of people that helped along the way. I want to, of course, thank David for all his support through the years I was in his lab as a postdoc. I really want to highlight Pat Houlihan, who's here. He is a co-first author on our paper. He did all the proteoliposome patch clamp work, and he actually did some recordings I didn't mention in of ORF3A by whole cell in a lung cell line, A549 cells that I generated, and we were not able to measure any attributable currents in that line either. Syl helped out with some biochemistry with SARS-CoV-1 or 3A and preparing samples for data collection. Tanya did initial imaging with SARS-CoV-1 or 3A and colocalization studies. Ben's contributions, he did the TEVC work, and others in the lab for helpful suggestions on manuscripts, on the manuscript, and as well, through meetings. The cryo-EM facility for data collection. I want to really highlight Zihang's his support, as well as Doreen's initial contributions. She came in during the pandemic and helped me out, especially because we could only be one person to a room during the earlier stages. And Rian, who did a lot of she helped collect a lot of the data toward the end when Doreen moved on to the NIH and started her own group. Sebastian Brauchi's group for contributions with the whole cell data, and Dajan's group for their work on the endolysosomal patch clamp studies. And then finally, yes, I'm at the University of Iowa. yes, I'm just starting my group, and yes, I'm looking for people. So, and this is becoming a standard comment by folks, but I certainly, if any of this work is of interest, or just ion channels and structure, and I have other projects that I'm thinking about too, feel free to email me at my email address there. If you don't feel comfortable emailing for some reason, you can always DM through X, Twitter. That's my handle. And I think that's it. So I'm happy to take any questions. Thank you very much. And thanks for the opportunity again to give this talk.
All right, thank you so much, Alex. That was really fascinating. And I see we already have a couple of questions here in the chat. So Damien Bell said, nice talk, Dr. Miller. Thank you. In your top 5 proteins with more than 2 TMs, the SARS-CoV-2 protein wasn't present. But does this suggest that the E protein, a putative viroporin, is not a viroporin, or just that it's not sufficiently expressed to make the top 5 proteins with more than 2 TMs?
Yeah, so I'll clarify here that this was an experiment that was done where we did not this is not an experiment using a SARS-CoV-2 virus. This is not during infection. This is overexpression of one of the SARS-CoV-2 proteins, and that was ORF3A. So the proteins we were likely to identify through mass spec. So I should mention it was overexpressed SARS-CoV-2 ORF3A purified and reconstituted into liposomes. So this is a sample that's been, highly purified and reconstituted. So, we were looking to see which proteins were we pulling down, during the biochemical multi-step biochemical purification. And so what you're going to identify here aren't, other COVID proteins. What you're going to identify are the of host proteins that might be, inadvertently being pulled down. In fact, actually, we did a strep TAG prep. So it's common to see mitochondrial proteins being pulled down. So it wasn't actually that surprising to see VDAC, in retrospect. But yeah, I hope that answers your question.
All right. So for the next question in the chat, Michael Young asks, hi, Alexandria, great work. I may have missed this, but it looked like you performed recordings at pH of about 7.2. Did you run any of your functional assays mimicking a more acidic luminal pH similar to what may be found in intact endosomes?
Yes.
Would this affect their ability to function as ion channels?
Yeah. So I didn't include this here. It's in our supplement, but Dajan's group did some recordings in endolysosomes, testing pH or proton, flux or conductance, and we weren't able to measure any currents in that situation. So it was done at low pH, at pH I think 4.5 or something like this. I've not tried to do any. I guess the subsequent question is, have we determined any structures at low pH but no, haven't tested that yet. It's a little bit difficult to do if you wanted to pursue that question. I guess the question would be is, either is ORF3A a proton channel, or is it, being gated, or, is it's conformational change dependent on pH, as an example. I haven't done any of those experiments. In nanodiscs, that's a little tricky because the MSP protein's unstable at low pH. Acetopsin actually works pretty well. Part of the reason why I went down that road, Acetopsin A is actually a protein that's expressed inside the lysosome, but you can use it as a scaffold protein too. So that's a direction we could go. Yeah, it's a good question though.
All right, so the next question comes from Carlos Villalba Galea. Is it possible that the pore could activate for interaction with a protein of the host, for instance, the ACE2?
Sure. There is certainly, we could be missing something, maybe that my counter argument to that would be that, all of the work that's been done to demonstrate that it's a viroporin has been done just by overexpression in, oocytes or in HEK cells. And we tried very hard to mimic those conditions, but, we could have missed something for sure. And you can't rule it out. Sure. Actually related question, could it be that we're missing it because we're not actually studying this during infection, right? Because there could be other viral proteins interacting with ORF3A that could be modulating its activity in some way. And sure, that's possible too. But I would say my argument to that would be that, the original data that supports that it's a channel was done by overexpression of one protein in the host cell, and we tried to mimic those conditions as well as we could.
Here's a question from Wang Zheng. Great work, Alex. I'm wondering whether the structure of SARS-CoV-2 or 3A resembles any other viral or host cell protein.
Yeah, it does. Is the short answer. So it turns out that OR3A is thought to be formed by a gene duplication event from from the M protein, which is one of the 4 virion proteins, right? So the M protein itself is, I guess it's the most abundant protein in the virion, actually. We focus on the spike protein, but that is the most abundant protein in the virion. Its structure was solved, I think, a year ago by a number of groups, and it looks pretty similar to OR3A as it was predicted, I guess, at the beginning of the pandemic, actually partially by AlphaFold predictions, as well as by, looking at evolution. But to my knowledge, the M protein hasn't been demonstrated to function as an ion channel. That said, could we glean any information based on based on its function, right? The M protein's function. So what we do know about the M protein is that, again, it's the most abundant protein, but as well, it appears to act as a scaffold protein. I'm not sure how strong the evidence of this is for this, but it appears to act as a scaffold for the nucleocapsid protein, which is bound to the RNA. As well, the virion. So maybe there's a scaffold or some other protein that OR3A is interacting with, which we haven't identified yet. That's certainly, again, back to the previous question, that's certainly something to consider. But as well, the virion I think it's 100 nanometers in diameter or something. It's pretty small. And so how is it able to maintain that membrane curvature? Another potential possibilities, the M protein facilitates that in some way. I guess there might be, and there's been some suggestions whether that, its interaction with the membrane, could it create curvature, and do you see, let's say, similar effects in the cell when you overexpress OR3A? And there is some evidence, preliminary evidence in the SARS and SARS-CoV-2 literature to suggest that OR3A could even be causing membrane disruptions and a mixing of organelles over the course of during late stage infection. So could it be that its main function is to disrupt membranes or do something or something to that effect? We don't know, but certainly it's important to think about this, and maybe they do have similar functions, but that's all I can say for the time being. It's a very good question.
All right, and then there's a couple of questions here from Sanju Lee. So she said, I'm wondering how highly these proteins are populated in your purified protein. And I'm guessing she's referring to the proteins that are co-purifying with OR3A. Have you thought about any specific interactions of 3A proteins with host proteins that are co-purified?
Yeah, right. So in terms of host proteins, I guess, that interact with OR3A, I guess what I can say about that is, I think a lot of the data that's been published has been done by overexpressing a viral protein in a host system. And I don't know if there's any data published where this has been explored during viral infection. What host proteins interact with OR3A during infection? And I think that's a really interesting question that hasn't been looked into. And we may be missing things by not looking at it in the context of infection, in the context of the 28 other proteins that this virus expresses. So I think that's a clear, an interesting question to pursue. But and then the first question was about the purification.
Yeah, so I think how highly, so I guess what is I'm extrapolating here, but I think what's the proportion of OR3A versus the co-purified protein that are in the sample? That's how I'm reading it.
I didn't, It's the majority of the population. I mean, it's a small. I don't have numbers in my head, but yeah, it's the majority of the sample. These are, I mean, I think the counts were listed in that table, but yeah, 90% of the sample. Yeah.
Well, we have a minute. I just wanted to ask you like a technical question. So you made a point of saying that you really needed to reproduce the lipid condition, the endosomal lipid condition for the nanodisk prep. Was that, did you find that the protein otherwise would crash out, or just didn't, it didn't resolve very well?
Yeah.
Or...
Yeah, no, that's a good question. So I'll start with, so I actually pursued this initially by both. Like I prepared, in fact, I think we have both structures in our paper where we reconstituted in the endosomal, lysosomal, lipid environment, and then the plasma membrane like environment, and they look very similar, right? The structures look very similar, and it had nothing to do with stability. It's just that by overexpression, we see OR3A in the endocytic pathway as well as at the plasma membrane. But I actually think that the plasma membrane localization is probably an artifact of overexpression. And so I think that's likely found in the endocytic pathway. And that is corroborated by data published years ago from Nevin Kurgan's group, where they did immunostaining rather than during infection rather than by overexpression. So yeah, that's why.
Wanjun says, thanks, Alex, for the answer. Just to know that we also tested ion channel activity or 3A in HEX cells and lysosomes. We got similar data as you presented. Great work again.
Oh, yeah, thank you.
And
Thanks,
there is another great work.
No, great. Thank you. And thank you all for attending.